Has the meeting started?
Just now I have started the live on YouTube. I'll be starting now.
Okay.
Let me share the link of YouTube to other people.
Yeah.
Good afternoon, sir. With your permission, I want to start my presentation on fungal allergen proteins. So here,
Hello.
So here we are going to talk something about the concept initially. My presentation would be on 3 parts. Basically, I'll be talking about what is allergy and how the concept of allergy has originated. And then I'll be talking about the various ways how the allergic extract for fungi will be made. And ultimately, I'll be ending with the testing how this allergenicity will be tested on the subjects, that is human people. So here, whenever we hear a term called allergy is usually a term, something which depict the pictures which are being portrayed over here, the drawings, where people will have running nose or irritation in the nose or something with upper respiratory tract. Mostly the allergy covers, almost many number of times allergy is covered as symptoms or a problem which is with the respiratory tract. And later on, people also found this will be even on the skin. So skin and respiratory tract are the major parts or major regions which are being affected with this problem. So before coming to know us about the allergy, there are few facts about the concept. This concept is something about immune system. To resist or protect ourselves from the various infectious agents, nature has provided all the animals with a system of protection called immune system. So this immune system was supported by, or it is supported by, cells of immune system called lymphocytes, or most of the cells which are present in the blood. This will be helping from the helping protection against the infectious agents. Here, the country, that is India, our country, we are proud to be the nation with second populous or second largest country after China in population. Maybe the numbers 1.3 or more, but we are second populous country in the world. And they told that there are the whole globe or whole world, the population in India is 20%. And they have told that there is a important allergic disease called asthma. Asthma, it is told that 37.5 million cases of asthma are there in India. And followed by that, there is the allergic rhinitis. It's a nose problem, which is being increasing day by day. The numbers are increasing. And this asthma is near about, when we are talking about 37.5 million cases, 50% of the cases are found in the children or school going age group or pediatric cases. And in most of the people most of the subjects or most of the children who are suffering with asthma are either under uncontrolled condition or under the severe condition. Similarly, this word allergy has come again from two pediatricians or child specialists. One is from Austria, other one is from Hungary. So these two people, Clemens and Belashik, these two people are the doctors or physicians by profession. They are doctors, allopathic doctors, who are known for their own field. Clemens is well known for a test done for detection of tuberculosis infection or a bacterial infection called tuberculosis, which is again a lung disease. So he was the one who started the tuberculin reaction, and Schick is also well known for his Schick test conducted or done on diphtheria affected people or to detect the diphtheria. So these two people are well known. Clemens and Belashik are known for designing or developing the test for different diseases. Diphtheria, again, it is a problem of respiratory tract, or we can say lungs. And Clemens is also known to have the test called tuberculin test that was designed by him. So these two people have coined a term called allergy, which has again origin from Greek words. Allergy is a typical reaction that is describing the process called hypersensitivity reactions. So here, these two people have given the term called allergy, and the Vienna Congress was held where these people have declared this terminology. So there are different type of allergies and their statistics. I'm going to discuss now about what are the allergies and what is their number of people being suffering. So it is a general allergy. We can see it is very well in the countries which are having number of industries. The industrial nations are having lot of allergy disease, and the range is again 40 to 50% are the children. School going children are being found to get the attack by the allergens. Then rhinitis. It has been told that rhinitis is a major issue in United States. During the year 2010 and 2012 data has been presented over here, where we can see 7.8% people are generally having roughly the rhinitis in the United States of America. And they told that the white children are more having the disease, when they say allergy of rhinitis, than compared with black children. So hay fever, it is very common among the white children than the black children. And they have told that this rhinitis is due to the sensitization, due to foreign proteins which enter the body from the surroundings, and IgE is the major culprit for the whole allergy process. Now, drug allergy. Drug allergy is also becoming very common. See, we have come to know that one particular injection was given to the patient and patient have become complicated. So such allergies are called drug allergies. Like penicillin, most of the people do not, or they do not tolerate the drug penicillin, or many more such drugs are there which are not being tolerated by the people just because of the same reason, that is the allergy. And they have told that near about 10% people around the globe are having the adverse reactions to the drug, either maybe oral or by injection. So The fatalities are also, death cases are also very high. It is up till 20% of the people are dying due to the adversity developed due to the injections or the ingestion of the drug. And majority of the diseases or the majority of the complications in the drug allergy are due to anaphylaxis. And the major issue is with the particularly food. General allergy we have seen rhinitis, we have seen the drug allergy. Now the major issue is found about the food. Even though there are people who cannot eat brinjal, some people cannot eat fish, or likewise each and every particular group of people are known to have their own diseases due to the food they do not tolerate. Intolerance, we can call food allergy, or also it is called the food intolerance. So they have found that again the data, 8% of the people around the globe are having the food allergy, and they have categorized again these food allergy into the particularly different age groups. So we can see that the more are among the people who are in the age of the 3 to 5 years. Food allergy is a major issue or major challenge faced in the age group of 9% people are generally having the food allergy are 3 to 5 years. Followed by that, we can see 14 to 18 years people are generally affected by the food. They do not tolerate or they have the allergy of the food. And the children who are having food allergy are not due to one particular food, but instead of one food, there are multiple foods which are causing them the issue. And not only food, after that we see that there is even some insects are also going to cause the particularly allergy. So they have told that the 50%, up to 50% of individuals around the world are having the fatal reactions in the particularly the insect bite or insect, whatever the insect things are, they like the stings, the mosquito bite. Also sometimes they may have the allergy on the skin. Likewise, and sinusitis is roughly 13% of the people are generally called to be affected by the sinus problem. That is also one type of allergy due to inhalation. And then skin allergy is most common among this is allergies, that they have found that the black children in United States were more affected with the skin allergies than white. And in Asia, where we fall, also 10% of the children were found to have the skin allergy. And among the skin allergy, the seriousness was due to particularly urticaria. And then followed by that, we are going to see once the allergies, when did this start, or when did this recorded, when this concept was first recorded. So when we see here the history of this allergy, what we have just seen statistically, here we have found even in the Greek period as well as in the Roman period also. But the study, the serious reports of allergy were recorded from the year 1800, and the person who is found in this picture is John Bostock. He was the one who noticed the hay fever as an allergic problem, and in 1869 the first skin test for the allergy was described or developed, where the pollen cut, the pollen was placed into a cut in the skin and watch for a reaction. So what this was done in the 1869 was they started testing the allergy by placing the pollen on the subject or the individual, and they made the skin a small cut and place the pollen over there, and they wanted to see what kind of development occurs. That was the first skin test, which is a crude form, which has been practiced in those times. And followed by that, in 1914, the therapy or treatment was in 1819. The first time allergy was reported, and it was 1914 where the allergy treatment was started, or the concept of treating the people to overcome the allergy problem was first started. And now, for treating the allergy, the better option they could find was using the antihistamine. Antihistamine was first developed in 1930 and treated for particularly overcome the allergic reaction. And later on in 1948, asthma was treated by using corticosteroids. And use of corticosteroids brought large relief or a big relief from asthma and allergy kind of reactions. So likewise, this was the concept where they have developed the end for the asthma or allergic reactions by using corticosteroids. They found at that moment that the mast cells are responsible for the particularly development of allergy. Mast cells will get degranulated, and the degranulation of the mast cells will develop the symptoms of allergy. In 1953, this was being developed. And later on, the story of immunoglobulins or antibodies was revealed, where the fifth antibody, that is called immunoglobulin E, was discovered in the 1963, and they have come to know that increase in the level of immunoglobulin E is responsible for the reactions or adverse reactions in the body. There are different types of immunoglobulins, like 5 immunoglobulins are there. Among the 5 immunoglobulins, the IgE was responsible for such adverse reactions in the body that is developing the allergy. And this immunoglobulin is responsible for the chain of reaction that results in the release of histamine or such kind of substances, chemical mediators from the mast cells. And this is again Professor Samuelson, who is one person who worked in the row of allergy, that is asthma and other inflammatory processes. He worked on leukotrienes and won the Nobel Prize in the field of medicine and physiology in the 1980. And now the question comes is, what is an allergic reaction? So when we talk what is an allergic reaction, it is nothing but when a person or an individual inhale or swallow something, or something comes and falls on their skin, top part of the skin, a sequence of events will undertake, and that will result in the particularly development of some symptoms. Like initially, when the body gets exposed to the antigen, that is, see, antigen is a substance that lead to initiate the reaction in the body, and this reaction may be, antigen may either lead to disastrous process or it may lead to protection. So the antigen, when it leads to protection, it is called immunity. When this go for devastating or disastrous kind of reaction, that is called hypersensitivity. Or hypersensitivity, one type is nothing but it is allergy. So here the people generally, after exposure to the antigen, that is entering in the body through either oral route or to respiratory route or injectable way, the antibodies are developed. And usually in a sensitized person, we have normal person and sensitized person. In a sensitized person, we can see that IgE comes to the rescue, and then this will these antibodies are present on the mast cell, and these mast cells are usually found in the respiratory tract and on the walls of intestines and any places they will be present. So the allergen or the antigen that binds the immunoglobulin E, and then it will release or it will cause the degranulation of the mast cell and release histamine, and that will be causing the symptoms of allergy. This is a process that has been shown how the antigens, these greener parts are nothing but this is the antigens, and this is a mast cell, and this is the degranulation process, and you can see the immunoglobulin E. So the binding of the antigen to the surface of mast cell, which is covered with the particular antibodies, will result in the particularly release of the histamine or chemical mediators that will result in the inflammation or results in the particularly the allergic responses. So allergen is any substance that could be an antigen either through oral route or through the respiratory route. It will result in the immune response that ends with the symptoms, and the symptoms, most common symptoms may be running nose or stuffy nose or irritation in the throat or itching in the eyes or sneezing, likewise these are the things. And as per the World Health Organization and International Union of Immunological Societies and Allergen Nomenclature Committee, there are totally 753 allergens, and these allergens are from different origin. And the high number of allergens are reported from the plant group. Like we can see Magnolopsida, it is 33%, and Liliopsida, it is 11%. So, and again, Coniferopsida, we have got 2%. So overall, here we can notice that 45% of the allergens are of plant origin, and we can see that 16% again comes from fungi, and the rest we can see the small number of the allergens are reported from the animal kingdom. We can see here the number is very scanty when compared with the plants. So here this is the data which has been published in 2018, and where the lot of allergens has been reported from the plant group than compared with the animal group. The arthropods among the animals, the arthropods are more notorious compared with the other group. The lesser number is here found with the other groups when compared with arthropods. As I told you, insect allergy is very predominant among the other allergies. So here 27% of arthropod allergens has been recorded by the Allergen Nomenclature Committee. So the symptoms could be like this, maybe more number of symptoms may also be possible, but here the symptoms, commonmost symptoms are generally the visible symptoms, where we can see the skin is get affected more. As if the respiratory system is involved, we can see there is particularly the sneezing will come, or the running of nose will be there. And if it is a food, we can see there is a disturbance in abdomen or stomach, bowel disturbance may be there, and sometimes either maybe more the regurgitation, or we can say vomiting may take place, or it may be generally the loose motions may appear. And again, insect allergies are again seen on the skin, like swelling or redness or pain kind of things can be noticed in the particularly the insect kind of allergies. And these are the again the exposure to the allergens we can see as individual, at home, in communities, and global changes. We can see these are the exposures where the society is getting for the allergens. So we can see at home, generally we can see that the dust mite or animals with fur are more unsafe compared here. Dog and cat are more important, and it is noted that there are at least 8 allergens have been reported from the cat. And mouse is also equally, the mouse skin or mouse whatever things are there, they are also very unsafe. Cockroaches are also very important in causing the allergy. And the last one is generally we can see fungi, which we are going to discuss today. So here again in communities also we can see the distribution, whatever the distribution is there. Again, same animals almost which are found in at home are involved in distributing the allergens to the subjects. And global changes, these are the reasons which are generally causing the wide distribution of the allergens, like climate change is there, then pollution is there, the different type of habitats are being disturbed, and that is also there. And the modern things which are used in the construction are also being important in the particularly distribution or making more allergens distribution in the society. And exposure to the major allergens from different sources. Here they have told what are the major allergens found from the different sources. Like arthropods, they have told that the feces of the arthropods are very important at the level of community or the level of the home. You can see that the different allergens are there and their sizes have been mentioned. So when we see the dust mite, it is again an insect which is majorly, its feces is of very small size, which is responsible for the particularly the allergy. Then in case of mammals, we can see cat, dog and mouse. All these are important. Like they have told that the source is in cat and dog, skin is most important source in case of the particularly allergy. And we can see that the saliva of dog, the saliva and the prostate secretions of the dog is again the major source. And in fungi, there are a lot of fungi, but here predominant fungi like Alternaria, Cladosporium, Aspergillus, Penicillium, their spores or their mycelial forms are found to cause the allergy. And equally, the plant pollens are very important as allergens. Like the tree species or the weed species or the grass species. So these are found, their pollens from the different plants are involved in the particularly source of the particularly allergy. So now talking to you about the fungi, they have told that the fungi or the large group of organisms which are found in the ecosystem, they have told that at least 25% of the population, living population is considered in the terrestrial, especially when we see the terrestrial ecosystem or terrestrial biomass, 25% of the biomass is covered by fungi, either in the form of mycelia or in the form of spores. So these are going to produce the spores, either asexual reproduction or sexual reproduction. So these spores especially are characterized as to be the allergy sources. So the fungi belonging to the phyla Ascomycota and Basidiomycota are important in the particularly one which are going to cause the allergic symptoms. They told that the airborne spores by fungi can travel thousands of kilometers from the origin of the source from where they have born. And they have told that the fungi are omnipresent and they are found in all the type of habitats, and they are more in number when compared in the air. When we check the composition of air, the spores of fungi are high in number when compared to pollen concentration, and the spores and the pollen will be variable seasonally. The seasonal distribution is a very important concept in case of the fungal spores and the pollen. So upon inhalation, the spores which are present in air or the fragments of hypha are known to cause the respiratory symptoms in the sensitized individuals, as discussed in the earlier part. These fungi are known to cause the negative impact on the airways or respiratory tract by producing different type of toxins, either the enzymes like proteases or some other enzymes which are hydrolytic in nature. And volatile organic compounds are most predominant by fungi in the damp environments. The damp environments where the old house or house not painted for a long time, which is having accumulated water in the ceiling or in the walls, will be responsible to develop the volatile organic compounds, VOCs. And they have told that the during the reports from last 4 decades, the allergic diseases are increasing remarkably from 10 to 30% in our country. And they have told that at least there are 80 fungi which are generally responsible for causing the airway diseases. And this exposure of allergens or fungal spores could be either indoor or could maybe outdoor. And generally, the unsafe concentration of fungal spores are 230 to 106 spores per cubic meters. And how actually the fungi are, how the fungi can be collected from the air. As I told you that outdoor spore concentration is more important in the study when we are performing the study on the fungi found in the air. And we will have the various type of samplers, and by using the different type of samplers we are going to collect the fungi or the study. Whenever we are doing the airborne fungi, we are going to use the different samplers, though there is a sedimentation method to collect the fungi by putting the open plate, and the spores may fall over the agar surface and they will be developed into the colonies. But here the different type of samplers have come, as not only the bioaerosols but also the chemical pollutants are also being recorded from the particularly air. So the sophisticated samplers have come in the different forms. You can see over here the impingers, impactors, which have their own different types of approaches to collect the spores in the air. And again, we can see the typical kind of, again, equipment where we can see the filters. We have the different type of membrane filters which will filter the air and collect the spores over there. And also we have cyclones where we have the tubes for collecting the particularly spores from the air. And the spore traps are more important and new kind of, I'm going to say, add up in the samplers. And even electrostatic precipitation is also a method where we can collect the fungal spores. So by collecting the fungal spores, the fungi are being characterized by microscopic method, and by seasonally collecting the fungi, they are going to record the they are going to record the fungal spores present in the air, and they are going to make a calendar that is, I think the size of the, I mean to say, the figure is small, but over here you can see the this is a literature collected from Day et al. In 2018, where they have recorded the fungi in different months that starts from July to August. So pre-monsoon, post-monsoon, that kind of data has been collected by them by using the samplers. and they have made the particular pictorial presentation or figure they have developed where they have found the various type of fungi over there. And they found that the now comes is after collecting the fungal spores, next we proceed for the particularly checking whether these spores or the spore developing fungi are safe or unsafe. Unsafe in the sense, I mean to say, here we are going to test whether they are really, I'm to say, causing any threat or allergy kind of symptoms. So maybe the normal person is not going to have the problem of allergy, but sensitized individuals who are having the asthma or any kind of such COPD or any, I mean to say, pulmonary diseases, or any people who are having chronic diseases, they may be at the risk. So to find out whether the people are, I mean to say, unsafe from the fungi are being collected through the sampler or sedimentation method, and then these fungi are cultivated on the agar media, and then after the development of the biomass, the biomass is harvested and proceeded for the particularly extraction of protein. The question comes, why only protein is the more important target over here? So there is even literature being given that the protein is actually a safe biomolecule, but there are several reactions like post-translational modification or such enzymatic activities, whatever the post-translational modification, or particularly the hydrolysis or proteolysis, or some subsidiary reactions which take place, will make the protein unsafe. So proteins are being found as to be the allergens when compared with other biomolecules. Though there are sugars, there are lipids and proteins. So when compared among the three biomolecules, proteins are found to be associated with the allergy symptoms in the individuals. So proteins prior to post-translation are considered as to be safe, but the post-translation modifications or proteolysis or any enzyme activities may develop, or they may have some kind of, I mean to say, changes, and like glycosylation or some kind of activities which modify the protein structure may be responsible for developing the allergy or hypersensitivity in the individuals. So as prior to this, I have shown you the spore calendar made by Day et al. In 2018, and they have studied the different type of fungi on the sensitized individuals, and they found that the different type of fungi like aspergillus species are found to be the allergic, and followed by that we can see fusarium species, and then carularia species are also equally found to be allergic. And now we are going to talk about the protein extraction. So protein extraction is usually done by the growing or cultivation of the mycelia. After the hyphal development occurs, the hypha are harvested from the culture medium, and then we'll proceed for the cell. We'll go for the hydrolysis of cell wall, and we'll collect the cell content, and we'll go for the salting out and salting in process. Either by salting in or salting out process, we are going to sediment the protein, or we'll collect the protein, and then we are going for the various sophisticated methods what we have been earlier taught by the earlier speakers. We have come to know that different techniques like SDS page, or we can see the affinity chromatography, or by using the IEF, that is isoelectrofocusing, we are going to purify the protein, and then we are going to separate this by using the Western blot technique, which has been already disclosed in the prior parts. And the purity of this will be checked by the ELISA, that is enzyme linked immunosorbent assay, will be performed for the protein. And then by using this methods, they are going to characterize the uh, protein extracted from the fungi. And then the next step comes is the storage of the fungal protein or allergens which have been extracted from the whole cell extract of the fungi is being taken. And the storage is major issue. The protein, as you know, the protein will be generally very, I'm going to say, it needs a special kind of storage practice. That is, lyophilization is more practiced for the protein. Whatever the proteins extracted will be there, the extracted proteins will be stored by lyophilization, and it will be for a prolonged period. It will be without losing the viability, it will be there. And the ultimate concept, the third part of my lecture will be about the how we will test the, how we will detect the allergy. So this is actually the part which is done by a specialist or a doctor who is treating such allergy kind of works, or allergy expert. So here we can see there are different type of tests which are available. One of the oldest method, as I discussed you in the beginning part, in the year 1896 or so, 1896 or so, we have seen that a pollen was placed on a skin of a person and checked for the reaction. Same concept, that is, has developed a skin prick test, where in the skin prick test it involves the particularly a drop of allergen is placed on the skin, either on the back or either on the hand. Generally on the hand it will be done. So here is, you can see the skin prick test performed over here, you can see. So if there are multiple allergens at a time, you will be done in a way how it is shown over there. And there will be two type of, I'm going to say, the controls are used. One is a positive control, that will be histamine, and the negative control will be saline, that is sodium chloride solution, 0.85% of sodium chloride solution. So the first one in the well, you are seeing the first one and the last one. The first one will be either, I'm going to say, positive control, and the last one will be negative control, or either any way they will be having that. And the two will be the allergens, or more than this, if the forehand is the target place, if the number of allergens are less. If number of allergens are more, then the back of the person, okay, the backside of the person will be used for the testing. And along with this skin prick test, there is another test is also there, that is called patch test. So patch test will be a little bit different from the skin prick test. So here mostly the preferable one is a skin prick test, where after the exposure to the allergen, the changes will be noted. That is in the form of wheal. Wheal is simple, nothing but it is a swelling, local swelling with redness we will be noting. And after the change, okay, after 15 minutes of the exposure or injection, or the injecting the allergen, the size is generally measured, and the shapes will also be generally taken. So here in this wheel, the different, I'm going to say, methods of measurement, like by using ruler or scale like a thing, it is ruler, it is nothing but scale like things, or vernier calipers also in laboratory conditions. Generally we'll be using vernier calipers also in the laboratory conditions. and the shape may be either oval or roughly round, or like some here the fluctuations may be there. And ultimately the way of measuring the wheel is being shown in the picture, D bit of the picture. And there are now the different methods have come where the accurate method, instead of using the ruler and doing the method, the scanning method of the particularly uh, the wheel will be done. That will be considered as to be the exact method have been developed right now. But it is particularly a little bit time consuming process. It is a time consuming process, and it is generally done in a sophisticated kind of conditions, when there is a lot of confusion about the patient. Sometimes the patient may may not be actually a sensitized person, and he may be allergic or not. That lot of confusion. When there is a confusion that patient's exact condition, health condition is not known, in such cases these methods are being used. And this is a size that will decide whether the person is whether the person is sensitive or not, whether particular person should whether he's allergic or not. That depends on the size. And the generally size as per the old scale and the new scale has been wheel measurement is been shown over here, where we can see the negative, mildly sensitive, moderately sensitive, very sensitive people are generally above 15 millimeter. If the size is at above 15 mm, then it is a very sensitive person, like that it will be interpreted. Maybe new type of data might have come right now, but the previous data is like that. And now as I told you just now, the after the person has come after the physician has come to the conclusion that allergy he's sensitive based on the wheel size and all, he will be suggesting the treatment. And if he is a respiratory, if the respiratory problem is there, then they'll be giving some decongestants and antihistamines, and that may be reducing the burden of running nose or stuffed nose or sneezing or itching. If not, the disease is not coming under the control, then they will be going for the corticosteroids. That will be the end for particularly the disease. So using of overdose of corticosteroids is also not safe. So the avoiding of exposure may be one of the most important part in the concept of allergy. So with this, I come to an end of my presentation. So if anybody is interested in asking the question, I am ready to answer. Thank you so much. Sir, over to you.
Okay, now it is unmuted. Hello.
Yes, sir.
Thank you, Dr. Ahmed. This is a very, very lucid, very clear lecture. I love your slides and the clarity of expression. Everything is very good. A couple of comments. The tuberculin prick test that you're talking, is it the Mantoux test that you're talking?
Yes, sir. It is Mantoux test. Today itself, I have seen that Clemens was the one who he was behind that.
Yeah. It is called Mantoux test.
Mantoux.
X is silent.
Mantoux.
That's okay. No problem.
Mantoux.
The second thing is, we have done a lot of work. There are many people in India who have done a lot of work on aspergillus allergens. We have published enormously on those things, collaboration with Dr. Usha Sharma. And also in Hyderabad, there is a doctor, Murthy. We have published with him also because he used to get us patients and all that. Mahavir Hospital here was collaborating with us. We have done a lot of work in the sense we have isolated those protein allergens, shown that they bind to surfactant protein D in lungs, and that's how they initiate a lot of immune response or so-called sensitivity responses. There is a lot of work on mast cell physiology. People have done how the histamine is released, why only IgE, why other antibodies like IgG and others, why they don't cause allergy. And the third thing is, these glucocorticoids are not advisable. I have a personal things to tell you this. We lost our daughter only because of that. So don't tell anybody that these glucocorticoids should be used. Never. It's very dangerous therapy. All right. I enjoyed your lecture. Very good.
Sir, actually, our work, what we did, myself and along with Manoharachari sir and my guide, Professor Mohan, what we did was we have done the extraction and we made the allergens and we have tested on the subjects at Kamineni Hospital with one doctor. And we have not gone till the treatment. We have just
No, just a word of caution.
Yeah, corticoids are unsafe, but here in that slide, it was meant to say what is
in Delhi?
The three parts I made in that presentation, that extraction part, and then
No, your slides are very clear. I want to add that in Delhi, there is something called Patel Chest, Vallabhbhai Patel Chest Institute and Hospital. They have a national referral center of all these allergens collected, both from cockroaches, flowers, and this kind of fungi and all that. There is a national repository, and they do undertake desensitization. Lakhs of patients come there. It's a very great institution, started long back. Avatar Singh Paintal, who was later Director General of ICMR, he was the director. He's an FRS, enormous work. My student, who is now a professor at Brunel University near London, has continued this work and has published enormously on aspergillus allergens. This is just for information, but I enjoyed your lecture. The slides are very good, very simple, very lucid, very good. Clarity is very good. That's how people should speak.
Thank you so much. Thank you, sir. Sir, now it is time for your lecture.
Okay. Initially, I want to apologize for something that might happen. I will start the lecture. Okay. Hello.
Yes, sir.
But in between, one visitor is coming, principal of a local girls' high school. In spite of my objection, she is coming to invite me for something else. I told her I'm speaking in a meeting and she doesn't listen. So anytime she may come and interrupt me, I will disrupt this meeting for 5 minutes, take the invitation letter and send her out. Okay?
Sure.
Yeah, please excuse me and bear with me for that. It's a St. Anne's Girls' High School in Hyderabad. All right?
Sure.
Okay. I'll start now by uploading the slides first. This slide visible?
Yes, sir.
Very good. I'm also audible, no?
Yes, sir.
Yeah. Now, friends, in a lighter vein, this is the last lecture. I hope you had a feast of lectures in the last 2 days, yesterday and today. This lecture should have come in the earlier slot, but because the other speakers spoke something and I'll have to continue that, I spoke on other topics. Now, this is a basic fundamental aspects of enzymes, how to understand them. So it is an introduction to enzymes. Most of you realize that biology, for about 1700 years, biologists did not do a single experiment. Subsequently, in the 18th century or later part of 17th century, some biologists started doing experiments. And these first people who investigated and did experiments were actually chemistry people. And these chemistry people therefore asked chemistry questions, but analyze living matter or living organisms for that. So the first question they asked was, the elements present in the inorganic material world, are these present inside a living organism? Very simple question. And because animals and others will protest, they analyzed only plant material. So they did the elemental composition and they found that the same elements which you see in the periodic table, they are present both in air, soil sample, as well as in living organism, say a flower or leaf or a plant bark or anything. Then they changed the question. They ask, the elements may be there, but the relative proportion, is it the same between inorganic soil sample and the living organism? When they did that, they got a surprise discovery that inside the living organism, that means all living organisms, the elements are there, but the relative proportion of carbon, oxygen, hydrogen, and one or two more are different from soil sample. Therefore, they said living organisms, and they did the compound analysis also. They said they isolated lot of things from plants initially, afterwards from animals also, and then analyzed these compounds. And therefore, in the early part of marriage with chemistry with biology, it was called natural products chemistry, because the products were obtained from nature. And in those days, nature means plants and animals, not the material nature. Subsequently, when they looked at these compounds, they were all compounds of carbon. And then they asked a very inconvenient question, can we synthesize these compounds? Because for the chemistry person, the ego satisfaction comes only if he can synthesize. And they undertook, but they failed. And then Frederick Wohler synthesized urea, a component of living organisms, from starting with silver cyanide, later ammonium cyanide, and showed that there is nothing mysterious about compounds of carbon present inside living organisms. No God is involved. I can synthesize. And that's how organic chemistry started. And then they asked a very important question. What are they doing inside the living organism? We don't find them outside. That means if you take a rubber plant and ask what is the role of rubber in a rubber plant, at that point of time in history, they didn't understand. So they asked, what are they doing? At that time, World War II took place. And if you remember, USA started making atomic bomb and then dropped it on Hiroshima and Nagasaki. Without going into detail, as a side reaction from this Manhattan Project, a lot of radioisotopes were made available to scientists. And some biologists like MD Kaman and others took these radioisotopes, incorporated them into synthetic organic compounds or biomolecules, and then injected them into organisms. And then they asked, what happens to this? Then they realized that all these compounds are not static, that they are constantly undergoing chemical reactions, and that If you inject A goes to B goes to C, all sorts of things are happening. This sum total of chemical reactions occurring inside a living organism, they called it metabolism. And each one of the biomolecule, they call it metabolite. But unlike material world or in the laboratory of a chemistry person, these reactions that you see within the living organism, there is not single uncatalyzed reaction. They're all catalyzed reactions. And what is the catalyst? A protein called an enzyme. And therefore, enzymes are called biocatalysts, without which no reaction takes place. Even dissolving carbon dioxide in water, which is what you see in Pepsi or Coca-Cola or any of those soft drinks, even that in our body is a catalyzed reaction. There is an enzyme called carbonic anhydrase, and that does the reaction. And therefore, people discovered thousands and thousands of chemical reactions in thousands and thousands of species of organisms, microbes, animals, plants, everywhere. And they called this as sum total is called metabolism. And all the reactions are catalyzed reactions. And the catalyst is called enzyme. Till 1980, these enzymes were all proteins. 1980, Thomas Seck discovered that ribosomal RNA can be an enzyme. Later, people discovered some antibodies can be enzyme, even though it is a protein. Normally, antibodies are not enzymes. And therefore, variety of enzymes like ribozymes, abzymes, and many others were discovered. We will today talk about only the proteinaceous enzymes. And as I said, how do we discover an enzyme? If you take a compound A and you incubate with a tissue extract, which is the source of the enzyme, at certain temperature and for a certain duration in a buffer of a certain pH, then you see formation of a compound B. Then you can make a safe guess that that tissue has an enzyme which converts A to B. And if you know the chemistry of A and the chemistry of B, you can immediately recognize what is the reaction. The reaction can be hydroxylation. Dr. Haleem Khan. Hello? Hello?
Sir?
Okay, నేను కిందికి వస్తున్నాను, మీరు ముందుకు రండి. Okay? Yeah. Hello, Dr. Khan.
Yes, sir.
I will disrupt for 5 minutes and come back.
Okay, sir.
That fellow has come, principal.
Okay.
Sorry, Dr. Khan, I have come back.
Sir, okay, sir. Don't say sorry, sir. Thank
I
you.
apologize for this because that principal came running to my house. Sorry. Anyway, we'll restart. If you remember, we were defining an enzyme as a biocatalyst, and we also told how people discover enzymes. Very simple. They expect a chemical reaction to be catalyzed by a tissue extract, an extract of a leaf or liver or intestine or root, anything. Enzymes are everywhere in all living organisms. What we have to learn, there is no single uncatalyzed reaction in our body or any living organism, E. coli to elephant. So while studying these enzymes, people ask many questions. First of all, they ask, what is the chemical nature of this? And a lot of work has shown that till 1980, all the known enzymes were proteins. You have already heard from Professor Yogendra Singh, what is a protein, what is its structure, and all those things. One should define reaction rate because enzymes catalyze reactions. There are some differences between a biocatalyst like an enzyme and an inorganic catalyst like palladium or platinum. Some of you have heard of Haber's ammonia production process, and that is the catalyst is a palladium, finely divided palladium. But in those cases, the catalyst does not become part of the reaction, and you need high temperatures and high pressure. But in the case of biocatalysts, you don't need all those conditions. And the enzyme also takes part in the reaction. There is something called active site, which we will explain. And therefore, enzymes as biocatalysts are different from inorganic catalysts that all of you are familiar in inorganic chemistry. Heat sensitivity of the enzyme catalyzed reaction. Please remember, I just now mentioned that inorganic reactions or any other reactions catalyzed by inorganic catalysts, they work at high temperature. That means they are thermostable. But in the case of biology, these so-called biochemical reactions, metabolic reactions, enzymes are extremely heat sensitive. That means if you go beyond 60 degrees or even 50 degrees, they don't work. And that is one of the diagnostic features of an enzyme, that it is a heat sensitive catalyst. However, if you isolate this enzyme from a thermostable fungus or any other bacteria or some such thing, you must have heard about PCR reaction in molecular biology. It is done with a DNA polymerase isolated from Thermus aquaticus, which is found in sulfur springs. And that enzyme is stable beyond 80 degrees or 90 degrees centigrade. Barring that, all organisms like us who live at 37 degree around that, our enzymes are stable only between 10 degrees and 40 degrees. Of course, if you lessen the temperature to 0 or sub-zero, they are stable, but they are not very active. They generate products, and to investigate, one should measure the product formed over a period of time. That is called generation of product versus time curve at different concentrations of the enzyme, the rate versus substrate concentration curve. These are some of the experiments that are done by people who have discovered an enzyme and who would like to understand the reaction. We'll explain everything. Supposing we take this example, glyceraldehyde 3-phosphate is a biomolecule. It is part of glycolysis metabolic pathway. It is converted to dihydroxyacetone phosphate by an enzyme called triose phosphate isomerase. Don't worry about all these long lamba chauda names. You don't have to by heart. Just understand them. So if you take this reaction as an example, we can do those experiments and find out whether the enzyme is thermostable or sensitive, whether the reaction velocity increases with temperature, whether the product formed increases with substrate concentration. Like that, one can analyze what are the conditions which will give me the best rate. Rate means dP by dt, which means amount of product formed versus unit time divided by unit time. So these were done initially, and the pioneering people who did this were Buchner, who studied fermentation, James Sumner, who Dr. Khan mentioned, that is the one who crystallized urease from jack bean. And then JBS Haldane, whom we hear very often in many areas. He also worked on enzyme. He is the one who gave us the rate equation as a steady state mechanism much 50 years back. Anyway, these are pioneers. They're always heroes for those who work in this area. Now comes the question, if every chemical reaction or biochemical reaction is catalyzed by an enzyme, and there are thousands and thousands of reactions from E. coli to elephant, then each reaction is catalyzed by a unique enzyme, and that enzyme you have to give a name, and therefore naming an enzyme became a very difficult task because people were confused. Let us say I am sitting in Delhi or Hyderabad and discover an enzyme and some reaction, then I give its name as I like. Similarly, somebody sitting in London may discover the same reaction, but he has given a different name. And therefore, to lessen the confusion, an international commission of enzyme nomenclature said that these thousands of reactions and thousands of enzymes can be classified properly. Remember, classification is a basic human instinct. You have read taxonomy in botany and all those things. Carolus Linnaeus, who gave us the binomial nomenclature, he knew the name of every tree, every plant in the world. And from that, we understand classification is a basic instinct whenever information becomes too large to handle. And so these people classified enzymes and reduced all the thousands of reactions, thousands of enzymes into only 6 categories. And as you read here, each category, number of enzymes come and they only catalyze this type of reaction. So there are only 6 types of reaction. However, how do you get thousands of enzymes? Under each category, you can sub-classify them depending on what substrate they are acting, what other features they have. So each enzyme gets a name, which is actually numerical name. That means 4 digits with dot in between. Say 1436, it means the first letter or first number tells you one of these 6 numbers to which group it belongs. Like that, you go by subgroup and subgroup and subgroup. The last digit will tell the actual substrate or reactant on which the enzyme acts. Like in the previous slide, it means it refers to glyceraldehyde 3-phosphate. And therefore, we can name any number of enzymes by only this classification method. And now it is very easy to remember them because classification simplifies not just in organisms, but also in enzymology. Okay, these are a data which tells you that many of these enzymes are simple proteins. However, there are enzymes which have an additional organic compound as part of the enzyme. And so the main protein is called apoenzyme, and the other organic compound is called a coenzyme. And depending upon how strong the affinity between these two, you call it coenzyme if it is loose, you call it prosthetic group if it is very tight. All right. So if you look here in this, there are a lot of coenzymes listed. You don't have to remember, you don't have to by heart. Majority of these coenzymes and prosthetic groups are derived from vitamin B complex, which means B complex, B group of vitamins, niacin, B12, all those things, they also work as coenzymes in many enzymes. You don't have to remember. Subsequently, you can read about it and remember the structures of all that. Many enzymes also have metal ions as the so-called cofactor. And some of them are, let us say they have copper. You can call it one example, cytochrome oxidase. In mitochondrial electron transport, which all of you have studied, both in zoology and botany, you see the cytochrome C oxidase coming at the end before the oxygen accepts the hydrogen and electrons, and that enzyme requires copper. That too in a +2 state. The valency state of many of these metals is multiple. Only one of the valency states will work as a cofactor. Similarly, you have a number of enzymes which require iron, which require potassium, which require magnesium, manganese, molybdenum, nickel, zinc. Urease, we have already mentioned, it requires nickel as a cofactor. Now let us come to nitty-gritty detail. What exactly is the velocity of a reaction or a rate of a reaction? As I said, very simple. Velocity is equal to differential of product formed per unit time. In algebra, you write it as dp by dt. D is a delta, which means smallest quantity of product formed per smallest interval of time. This is called a differential equation. You can write this as this, or you can also write it as with a negative sign, delta S divided by delta T. What does this mean? S means substrate. If A goes to B, A is called a substrate, B is called a product. Therefore, these are designated as S and P. And so you can have a positive sign and call it a dP by dt, or you can have a negative sign, call it minus ds by dt. This defines the rate of a reaction. These two people, Professor Michaelis and his technical assistant, Maud Menton, a brilliant lady, they investigated enzyme reactions by changing conditions, which I have already mentioned. X-axis, you will have the substrate concentration. That means A. So if you read this, therefore, it says that the velocity is proportional to the substrate concentration.
Sir,
hello?
Share screen has gone.
Sorry?
Your screen is not visible.
Something else came here. Can I close that?
Here, your screen is not visible.
So what do I do?
You reshare the screen. Stop sharing and again reshare.
Okay. Is it visible now?
No.
Okay, I will start again. All the bottom, all that has disappeared. I think I may have to close it and restart.
Okay.
Somebody has to let me in.
No, sir. The meeting is.
Those bottom toolbar is not visible. That's why I am not able to upload. I have sent these slides. Can you project it?
Yes, I'm seeing that only.
Yeah, okay. Can you see now?
I am sharing now. You are sharing?
Okay, you are sharing. Okay, very good. So go to the next slide. You can hear me, na?
Yes, sir. You are audible.
Very good. Very good. Thank God. All right, next slide.
You're talking about Michaelis and Menten.
Yeah, the next we have crossed this. Yeah. Therefore, you see that this is the result they got, and it has now become a famous curve. This is a rectangular hyperbola. What you see is the velocity as a function of substrate concentration. People thought it is a linear, it is non-linear. Incidentally, I will tell you that in biology, all responses are non-linear. Whatever be the bacterial growth curve or population increase, all sorts of things, everything is non-linear. This rectangular hyperbola, as you see, velocity increases very fast. Subsequently, it becomes plateaued. And if you draw a tangent asymptotic with the upper part, and it cuts the y-axis, that gives what is called as maximal velocity. And if you take half of that and draw a perpendicular on the rectangular hyperbola curve and drop a perpendicular from that cross point to the x-axis, you get another parameter called Km. And as the x-axis represents concentration, Km also the expression is in terms of concentrations, moles per liter or whatever the unit you have given. Next slide. And therefore, enzymes catalyze reactions in which there is a velocity and the substrate becomes a product over a period of time. Now people asked, what is the reaction pathway? What happens actually? And to make us understand, this Michaelis and Menten wrote that pathway in the following terms. Enzyme plus the substrate becomes a complex called enzyme substrate complex, and that dissociates into free enzyme and a product. Now this looks very simple, but it is very, very complicated idea. When you see free enzyme after the formation of product, it is not same as the first free enzyme which we started with. It has undergone conformational alterations. Therefore, after some time, you don't see the reaction. The enzyme becomes tired, it becomes denatured, it is dead. Now, in the first part, you see that enzymes substrate combine to form ES complex. What made people think that there is a ES complex intermediate? Because if you look at the earlier velocity substrate curve, it was plateauing off after increasing. If you increase the substrate and if there is no other limiting factor, that curve should have been straight line, but it is not. Therefore, Michaelis and Menten predicted there was a limiting factor which controls the velocity, and that limiting factor is the concentration of ES complex. Because you added limited quantities of enzyme and you only change the concentration of substrate, after some time, all the enzyme is saturated by the substrate, and therefore there is no more ES complex formed to give you reaction, and therefore increasing substrate concentration does not allow increase in velocity. That is indicated in this slide. Next slide shows you that this is precisely what is happening. Of course, we came to know this after 50 years when people did the X-ray crystallography of enzymes along with the substrate. They discovered that there is something called active site on the enzyme surface in which the substrate goes in and binds. So this is a picture which shows that the substrate is binding to the active site of the enzyme. What is this active site? 5 to 6 amino acids within the enzyme protein. They may not be linear neighbors. They may be on different portions of the enzyme, but they come together because of the folding of the enzyme protein and form like a gufa, a sort of a well in which the substrate can bind and form the complex. Please remember, this is not a covalent complex. It is other forces, ionic, hydrophobic interactions, all sorts of things will come. Next slide. And this, the chemistry people told us long back that for any reaction, you can plot free energy change versus x-axis reaction coordinate. Even today, nobody tells you what is this reaction coordinate. It is not time, it is not concentration, it is nothing. No textbook will tell you the meaning of that. And they predicted that this reactant A undergoes some transition and it becomes a transition state condition in which it has undergone distortion and then it falls to a ground state. And that active state or the transition state is the reactive state of the compound. Same thing is supposed to form in the enzyme catalyzed reaction. The ES complex, the substrate is held by the enzyme in this kind of an excited transition state. Next slide. Same thing is shown here. Next. So if you were to try to understand over a period of time, when I mix an enzyme and a given substrate, and over a period of time monitor some of these, you get this kind of curves. The substrate will go down in concentration, that is shown. The enzyme substrate complex will increase in concentration initially and then remain constant, where you can say DES by dt is 0. The free enzyme concentration initially falls and then again becomes steady state. The product increases over a period of time, as is also shown. So these are all what we call kinetic picture of the reaction. It indicates the reaction pathway. Next. So if you were to do this, don't get scared. This simply says the whole thing in nutshell. Velocity is proportional to ES. Therefore, you get that rectangular hyperbola. We understand. And now if you put that totally and express the rate equation, E plus S forms ES and the reaction rate constant is K1. Every reaction is reversible. So the reverse reaction, ES complex, dissociating into E and S is rate constant is K2. And then the ES complex becomes E and product, that is rate constant is K3. Now, if we do some algebraic jugglery, we don't have to worry about how all these things, the biochemistry students have to derive, but don't worry about all this. We'll go to the final equation next. That is what written at the bottom. Velocity is equal to maximum velocity multiplied by the substrate concentration divided by Km plus substrate concentration. It's very easy to derive, but I don't want to bore you with all that. Therefore, this is called the rate equation for any enzyme. For a long time, people thought that all enzymes behave like this, so they were all called as Michaelis enzymes. Subsequently, people discovered that there are many enzymes who don't follow this kind of a thing. They give you not rectangular hyperbola, but a sigmoidal curve. Oxygen binding to hemoglobin or any other enzyme, there are many enzymes, you don't get rectangular hyperbola. So the rate equation will be different there. I will not confuse you with all that. We don't have to understand. Just understand that for Michaelis type of enzymes, which will give you an enzyme substrate figure as a rectangular hyperbola, this equation applies. Next. There are compounds which will increase the rate. These are some examples given here. But the point is, what is the maximum rate that an enzyme can catalyze and what is the minimum rate? One of the characteristic features is that enzymes increase the rate of a chemical reaction thousands and millions of fold. You see on the right side, figures are given as 10 to the power of 5, 10 to the power of 7, and even 10 to the power of 17. So there is an enzyme called orotidine monophosphate decarboxylase. It acts on orotidine and removes the carboxyl group. And the rate enhancement that you see is 10 to the power of 17. There is no catalyst in the inorganic chemistry field which can do this level. That's why we say enzymes are highly efficient. Next. All right. How is this enzyme activity regulated? That means regulation of Km and Vmax. Next. Next slide. So there are many factors. One is a pH. If you take 4 or 5 enzymes and assay them at different buffers with different pH, you will get the activity versus function, the pH, and you see different type of curves. Some of them are bell-shaped curve, where the peak you see around pH 7. So there is a glucose 6-phosphatase will give you this. Trypsin also will have pH 7. Whereas if you take another enzyme, pepsin, the pH optimum is in the acidic range, 1.8, and that's how the curve is shifting to the left side. So different enzymes have different pH optimum, and therefore it says that pH influences rate of chemical reaction, that is biochemical reaction. Next slide. Next slide. These are different types of curves. You don't have to worry about it. It shows that there is a variation in pH optimum. Next. So if you do that same enzyme reaction versus time, that means you start the reaction and you look at your stopwatch and then keep on measuring the product at different time intervals, you will generate this kind of a curve. And what you are seeing here is the time course of a reaction at different concentrations of substrate. And you notice the curves are increasing in slope when you increase the substrate concentration. All right. Next. Now, for biologists, whenever we see a curve which is not a straight line, taking reading in between is a very difficult. Looking at a rectangular hyperbola or a circle or anything, pinning down at a particular point and drawing a tangent is difficult. So what people have done is to manipulate the curve. It means if we have a rectangular hyperbola, can we make it straight? There are many ways of doing straight. This is basically algebraic manipulation. One way is you take logarithm of every item that you got value, and then you see the curve actually contracts, and therefore even a S-shaped curve will become straight line. This is called log-log plot or semi-log plot or something. Another example I'll give you, most of you might have seen a French curve. It is made of soft lead. You must have used it for drawing. And there, if you have this curve and you want to make it straight, you hold the two ends and pull it. Exactly, algebra will do the same thing. Instead of pulling, it is contracting. Therefore, any curve will become a straight. So this is one method where you plot reciprocal of the velocity 1 by V0 versus reciprocal of substrate. Earlier we saw Vs curve, which is rectangular hyperbola. Now if you plot it 1 by V versus 1 by S, you get a straight line, and the line cuts the x-axis on the left side, negative side, and that is the value of minus 1 by Km. So we can easily get the value of Km. And the intercept on the y-axis represents 1 by Vmax. So the reciprocal of that gives me the maximal velocity. So reading a rectangular hyperbola is difficult, whereas reading this curve is very easy because it is a straight line. These are all manipulations done by the scientists who work in this area. Next. Next. Now, what else do enzymologists do to understand? They want to purify, that means homogeneous form of the enzyme from plant extract or liver extract or brain extract or wherever the enzyme is found. You can purify them using separation techniques. Dr. Khan showed some chromatography columns and affinity techniques and all. Using the same, including electrophoresis, variety of electrophoresis, precipitation methods, one can purify enzymes. I will not go into detail. When you purify an enzyme, there is some property called specific activity. How much activity you get per milligram. That value, if you can calculate and plot it versus the different steps of purification, you will see that specific activity increases, and then when it is pure, it will not increase anymore. It is constant. That means all the protein that you have in your hand is represented by the enzyme. That is the meaning of pure enzyme. It has nothing to do with holiness. Pure means homogeneous. And the specific activity should increase and remain constant. That is a marker to say that I have purified. Next slide. So if people were to do this on variety of enzymes and they determined the Km, and that I gave you some examples here, you notice that the Km values range from 0.4 millimolar to 108 millimolar for chymotrypsin, which means there is a wide range of numbers which will characterize the Km value for different enzymes. There are 10,000 enzymes, let us say, all of them will have different Km value. Next. Next. Next slide, please. Now, if you now derive a property called Kcat, which means Km value divided by so-called Km, velocity, maximum velocity divided by Km, you get another thing called catalytic efficiency. And that is given for different enzymes. And you notice here, these are some of the most efficient evolved enzymes: lactamase, crotonase, catalase, acetylcholinesterase, carbonic anhydrase. And if you see the turnover, that means if you take carbonic anhydrase, 1 million molecules of carbon dioxide can be dissolved in 1 second. That is the efficiency. The Kms are given, and what you notice is Km value has no correlation to the efficiency of the enzyme. Next. These are different type of, when you look at the rate and plot this rate curve, then you get this kind of a thing. If you say first order curve, like a radioactive decay, you see that rate keeps on changing, and therefore they calculate something called half-life. Radioactive decay is a typical example. Any protein decay is another example like that. So every one of the enzyme reactions, people ask what order of the reaction it represents. Is it zero order? That means the rate is independent of substrate concentration. Is it first order? It means the rate is dependent on the substrate concentration raised to the power of 1. Is it second order? Which means the power to which it has to be raised is 2. Like that, majority of enzyme catalyzed reactions are basically first order, but there are many which catalyze second order reactions when there are more than one substrate. Next. Now, if you take enzymes, most of the Michaelis enzymes have one substrate. So it is very easy to understand and write the reaction pathway, understand the mechanism, as well as construct the rate equation and other thing. But there are examples of many enzymes which work on two substrates. That means S1 and S2. What will be the reaction pathway for that? They have to form, of course, ternary complex. Enzyme reacts with S1, first substrate, forms a ES1 complex, then picks up the S2, the second substrate, becomes ES1, S2 complex. This path can also be different. It can pick up S2 first and then go to S1, S2 complex. This is one way of doing it. This is called ordered sequence. Of course, there are many other examples where ternary complex is not formed. The enzyme picks up one substrate, forms a product, goes back and picks up the second and then forms a product. This is called ping pong mechanism, because this is like some of you have played table tennis, and this ball goes between the two players. Exactly same mechanism happens. The substrate is shuttled between the two forms of the enzyme. That's why it is called ping pong mechanism. Next. Next slide. Yeah. Now there are many small molecular weight compounds, biomolecules, which can inhibit enzyme, and including Michaelis-Menten, many other enzymologists studied this phenomena of inhibition of an enzyme by a compound. It is very important in medical practice because many crucial enzymes can be inhibited, and so the disease symptoms can be reduced. And therefore, people study what is the mechanism of this inhibition. In one type of inhibition, this is called competitive inhibition. The compound resembles the substrate in structure. Therefore, it tries to occupy the same active site. And so the two compounds, the substrate and the inhibitor, try to bind to the free enzyme active site, and therefore the total rate will come down. Because ES complex is a productive complex, whereas the EI complex is not a productive complex. All right. And therefore, this is known as competitive inhibition. We can identify this and realize it by having those reciprocal plots. Some of you might have done. Next slide. I showed you this curve, which is a reciprocal plot where 1 by V is plotted against 1 by C. Look at this curves. There are 3 curves known. And if there is no inhibitor, you have the first curve. If the inhibitor is more, then you have the second curve. That means the curve is becoming, slope is increasing. And if you have more of the inhibitor, the slope increases further. If you look at the bottom x-axis on the left side, you notice that as the inhibitor increases, 1 by Km decreases, which means Km is increasing. This is called competitive inhibition, where the Km value increases, but the Vmax comes down. Next. In addition, there are other ways. We will not bore you with the detail. There are non-competitive inhibitions, uncompetitive inhibitions, and all. Things will get complex for you, but this is good enough to understand. All this points out that there is an active site in which this kind of a thing is happening. But in the case of non-competitive, it is not at the active site. It is another site, and that is called allosteric site. Some of the enzymes have allosteric sites, and compounds bind to that and inhibit the enzyme. That is called allosteric inhibition. The grandest example for which a Nobel Prize was given was hemoglobin, as you know, is a blood pigment which is present in our erythrocytes. It binds oxygen in the lungs and then goes to the tissue and delivers the oxygen. This is compared to an enzyme binding the substrate and giving up the product. And Jacques Monod of France studied this in detail and gave the kinetics of that. And he discovered this phenomena of allosteric inhibition and activation. 2,3-bisphosphate glyceraldehyde is an allosteric inhibitor. Not only that, if you go up the Himalayas, as you go up and up, your breathing becomes very problematic because of less amount of oxygen. And that is because this compound increases in concentration, binds to hemoglobin, does not allow it to bind oxygen. That's why we have breathing problem when we go up Himalayas or Mount Everest or anywhere. Next. All right. This can also be discovered by reciprocal plots. We'll skip this. Next slide, please. These are all other types of inhibition. Don't worry about all this. It becomes more and more complicated, but learn the basics, that is enough. Next slide. Same thing is shown here. Next. So if you look at this, then people asked the question, can we inhibit this active site by some chemical modification? Yes, you can do so. This is a diisopropyl fluorophosphate, which can inhibit some proteolytic enzymes by actually forming a covalent bond with the active site serine residue. Next. Of course, these follow typical reaction mechanisms that you study in organic chemistry, nucleophilic reactions, electrophoretic substitutions, all these things are allowed. Therefore, the compounds which can do this, they are called as nucleophiles or electrophiles. Some examples are given. You don't have to worry or by heart any of these things. Next. This is some of the mechanisms given, and this is penicillinase acts on penicillin acts on bacterial cell wall, and you can have an enzyme which is called penicillinase, which degrades penicillin and therefore make it inactive. So this is actually a transpeptidase reaction. We will not worry about it. This is about penicillin action. This is an example where an antibiotic is inhibiting one of the enzyme catalyzed reaction. Next. This is for other type of reactions. We can skip all this. This is good for biochemistry students, not for general biology students. All right. But what you have to remember is that those of you who have done some chemistry realize that there are what are called reaction mechanisms, and there are 4 or 5 types: acid-base catalyzed, nucleophilic substitution, electrophilic addition, and all that. Same mechanisms also are working for biochemical reactions, that is, reactions which occur inside our body, what we called as metabolic reactions. The reaction mechanisms is the same thing. This is an example of a keto-enol tautomerization, which is general acid catalyzed. Same thing occurs in our body. There is no difference. Next slide. These are examples for different types of reactions: acid catalyzed, base catalyzed, and all that. Next. I will spend a few minutes on a different type of enzymes. These are called allosterically regulated enzyme. Till now, we were looking at curves which will tell us that enzymes or velocity is regulated by either substrate concentration or temperature or pH or inhibitor, like that. But now we will look at some other enzymes which are inhibited by compounds which don't resemble the substrate. So you don't see competitive inhibition. This is called allosteric inhibition or allosteric regulation. First, Jacques Monod, a French biochemist, he studied this. He also studied lacoperon in microbes, in E. coli, and he told us the details about this allosteric regulation, which is given here in the form of a cartoon, where the enzyme which exhibits this is supposed to have more than one polypeptide. Therefore, here is an example which is having 2 polypeptides. One is called catalytic subunit, C. Another is called regulatory subunit, or R. Now, any enzyme which has got this multimeric protein binds to both the substrate as well as the allosteric compound. So if it is that compound is a positive modulator or a negative modulator, it does not matter. It binds to only the so-called regulatory subunit, induces conformational alteration, and therefore the catalytic activity either comes down or goes up in rate. That is what is indicated here. Next. Next slide. This is given in a so-called space-filling model of 2 conformers of the enzyme, the T state and the R state, which I have already described. The T state is kinetically inactive, the R state is active, and there is always an equilibrium between the 2 conformers of this allosterically regulated enzymes. Although this is not an enzyme, hemoglobin is the best example to understand allosteric enzymes. Glyceraldehyde 3-phosphate dehydrogenase is an allosteric enzyme. There are many examples. Carbonic anhydrase is an allosterically regulated enzyme. Okay, next. This is the curve that I was telling you, that if you were to assay an allosteric enzyme, you will get a sigmoidal curve, not a rectangular hyperbola. But we can convert a rectangular hyperbola to sigmoid or sigmoidal to hyperbola based on the allosteric compound. If it increases the affinity, it becomes T is converted to R state and you get a rectangular hyperbola, as you see in the blue colored curve. On the other hand, if you have allosteric inhibitor, it converts the R state to T state. So binding to substrate is made more difficult. Therefore, you see now the curve is shifting to the right side. It almost becomes straight. And therefore, this sigmoidal curve is only a transitory curve. We can convert this to rectangular hyperbola or a straight line based on what is the allosteric regulator compound. Next. This is different shapes of the same enzyme curve based on the concentration of the allosteric compound. You see this in the hemoglobin case, when you increase the concentration of carbon monoxide or increase the concentration of 2,3-bisphosphoglyceric acid, you will get this kind of different curves from straight line to rectangular hyperbola or from rectangular hyperbola to sigmoid. All these can be derived and experimentally demonstrated. Do not bother about detail, but try to understand that this curve is only generated by the scientist, which indicates the kind of behavior, but we can convert any curve into any other curve by altering the concentration of the allosteric compound. Next. Many a time, the enzyme undergoes covalent modification. Yes, today morning as well as yesterday, we discussed phosphorylation of many enzymes activates the enzyme. This is called regulation of enzyme activity by covalent modification. One example is where phosphorylation affects. You must have heard of an enzyme called glycogen phosphorylase, which degrades glycogen in our liver and muscle. And that enzyme exists in two states, phosphorylase A, phosphorylase B. The B is inactive, A is active, and B is converted by phosphorylation, covalent modification into A. That's how the activation occurs. I gave another example, phosphorylase B kinase in the morning, where by phosphorylation, you can activate an enzyme. By dephosphorylation, you can inactivate an enzyme. This is one type of regulation by covalent modification. There are hundreds of examples. Next. This is another example of acetylation. You have studied chromatin structure. There is a major protein called histone, and that histone can undergo modifications like acetylation, and that regulates the histone acetylase or deacetylase enzyme. And therefore, this is another chromatin modification leading to gene expression activity change. Next. Further examples, ADP ribosylation. We don't have to worry about all the details. These are a variety of examples. In the morning lecture, I said there are 240 modifications, post-translational modifications. All these are post-translational modifications. 240, don't try to by heart. Remember one or two, that is enough. Next. Next. And therefore, covalent modification like ubiquitination, acetylation, phosphorylation, methylation, all sorts of things, they regulate the enzyme activity or velocity of enzyme catalyzed reaction. Next. This is a bacterial cell wall, which is a N-acetyl glucosamine, N-acetyl manosamine alternating polysaccharide structure. And this can be degraded by respective hydrolysis or carbohydrases. There is a lot of work on this. Penicillin actually is the one which blocks the biosynthesis of that and therefore works as an antibacterial compound. Next. This is a figure, three-dimensional structure of lysozyme, tertiary structure. Lysozyme is found in our tears. It can also break bacterial cell wall. And this is the shape of that enzyme. Next. There are a number of enzymes which are called proteases, which means they degrade peptide bonds. So they are proteolytic in their activity. Hundreds of varieties are available. You don't have to by heart all this, but try to remember trypsin, chymotrypsin, because they play a part in digestion in higher animals. Next. Okay. We'll not go and skip this because this is detailed mechanism of reaction by serine proteases like chymotrypsin or trypsin. We don't have to worry about all this. Next. What is important for you people is that this study of enzymes is not simply esoteric and is of interest only to the biochemistry people. Many diseases are characterized by finding certain decrease in enzymatic activity or increase in enzymatic activity. And if it happens to be an enzyme from a tissue, then, for example, some liver enzymes like tyrosine aminotransferase should not be found in the blood. But if the doctor finds it in the blood, that means there is a liver damage. And therefore, Enzymology is also useful for diagnostics. Supposing pancreas has an enzyme called alpha amylase, it should not come out in the blood. If it comes out, and that means you have pancreatitis, inflammation, or even pancreatic cancer. So one has to take discovery or finding the enzyme in a wrong place shows that there is an underlying physiological disorder or disease. Therefore, a lot of things have been developed, put together as enzyme technology, where enzymes are used as an immobilized form or some other form as a diagnostic tool. We'll take some examples and then we'll close the thing. Next. I have already talked about this. In some of the diagnostic procedures and in purification, large scale, industrial scale procedures, these enzymes are immobilized by any of these four methods. In the morning lecture, I gave details about all these things. Next, if you use that, then you can purify or conduct the reaction in a large scale fermenters where immobilized enzymes are used to produce products in kilogram quantities. Normally, when we do enzyme reaction in the test tube, we only get micrograms, but you can do it in kilogram quantities in bioreactors and fermenters if you do immobilized enzymes, and that becomes an enzyme technology. Some other occasion, we will discuss how to make enzymes thermostable. That is also part of biotechnology. And some other occasion, some other college, I'm talking about it. Let us not worry about it now. Enzyme pathology says that you can use this enzyme presence or absence or amount of present for diagnosing pathological conditions. Therefore, it is called enzyme pathology. Next. Next slide. One example is you have looked at this structure of hemoglobin, which is a tetramer, and we have already discussed, probably not in this lecture, but there is a disease called sickle cell anemia, where in hemoglobin there are four chains, two types, alpha and beta. In one of the chains, one amino acid is replaced by another amino acid, a lysine by leucine or vice versa. Then the conformation of the hemoglobin changes. And when the conformation of the hemoglobin changes, the erythrocyte shape also changes. The erythrocyte now becomes sickle shaped, and that does not bind oxygen properly. And so these people will have anemic condition because of lack of certain critical amounts of oxygen in the body tissues. Similarly, there are other alterations in the amino acid sequence or position in hemoglobin, and they give rise to a number of diseases. They are called as hemoglobinopathies. Thalassemia is one great thing which is fatal. I will not go into detail. Like that, there are a number of diseases where a particular protein or an enzyme, in most cases, becomes pathological or leads to pathological changes. Next. Next. So if you see that sickle cell anemia or any other change in pathological change in hemoglobin, the oxygen binding curve changes. Here is a graph which will show you that myoglobin also is an oxygen binding property, but it is in the muscle, it does not move, and it does not serve as an oxygen carrier for our body. Whereas hemoglobin is in erythrocyte and it moves in the blood when the blood circulation comes and goes to the tissue and releases oxygen. And this binding and oxygen release is characteristic of hemoglobin, which can do the function only because the velocity substrate curve is sigmoidal. The meaning of this, I don't have time to explain to you. We will have to calculate algebraically the Km values and the dissociation constants, and we will then understand because of the sigmoidal curve and the result from that, this hemoglobin binds oxygen in the lungs, but releases oxygen in the tissues because of the presence of carbon dioxide, which allosterically regulates this enzyme activity and makes this curve steeper. Therefore, it releases oxygen. This is the physiological significance of this allosterically regulated sigmoidal velocity substrate curve. Next. This is the compound I refer to. which is a 2,3-bisphosphoglyceric acid, which is an allosteric inhibitor of hemoglobin binding to oxygen. Next. I already talked about sickle cell anemia, where there is a single amino acid change in the hemoglobin chain, and that leads to the erythrocyte shape alteration and less binding of oxygen. Therefore, it is a disease. In thalassemia, there are a number of mutations. There are hundreds of varieties of thalassemia. Each one is characterized by a particular mutation in the hemoglobin chain, and that leads to pathological condition. You need blood transfusion, and it's a fatal disease. It affects childhood, and Queen Victoria had in their family this kind of a disease. Next. So I will close this. What I have done is to give you a cursory account or a bird's eye view of this whole field called enzymology, which is useful not just for biochemistry, it is useful for every area of botany or zoology, physiology or genetics. In every case, medical practice, diagnostics, prophylaxis or therapeutic. For example, if I am as an old man, if I can't digest food properly, we will take tablets which contain trypsin and chymotrypsin. Therefore, enzymes can also be therapeutic agents. And therefore, we have given a survey of the discovery of enzymes, the reactions they catalyze, the classification of enzymes, enzyme nomenclature, enzyme reactions, mechanisms, and where are these enzymes useful in biotechnology, in medical practice, and in variety of other basic chemistry and basic biochemistry. I will close the lecture at this point and take any questions, any number from the audience. Thank you, Dr. Khan.
Thank you, sir. Participants, please, if you can put the questions in chat box, we will be reading out and getting the answers for the questions. Sir, I have one question.
Please, please.
Sir, these digestive enzymes are sold in the form of syrup or sometimes in a capsule form.
Yeah.
How do say these enzymes are going to work? Actually, in the in vivo system, the enzymes are produced in a trace amount and their concentration may be quite different. But how this supplements are given for digestion repair or something like that?
Yeah. See, inside the cell or even when they are normally secreted during digestion, no doubt they are in small concentration. Please remember, increasing the enzyme concentration has no deleterious effect. Because we are taking tablet or capsule, they are covered with a gelatin layer so that they are not themselves digested. Please, mechanisms for that. It is usually found as a trypsinogen or an inactive form. It is activated by mild proteolysis. Same way, when we take a tablet, we are protected from its digestion by the pancreatic trypsin by certain layer of coverage. But net effect, because it is given in large doses, it can digest the food, the proteins in the food that we have ingested, because our pancreas is not able to excrete or secrete enough quantities of trypsin and chymotrypsin.
And I had a question since yesterday that nowadays the people who are going for gymnasia and making the body, they are using a lot of protein supplements, and the protein supplement source could be synthetic. And can you highlight this concept?
Yeah.
About how this protein supplements are going to, either they are going to show the ill effect, or what could be the
Number one, these are some of the general statements I am making. In all diet, we need a certain level of protein to ensure proper amino acid and other things that are required for muscle building and all. Therefore, pure vegetarian diet which does not have any protein, plant proteins are not good, animal proteins are good, milk proteins are good. And therefore, some people who are in the name of religion don't take any non-vegetarian food or even good milk protein. If you ask Maneka Gandhi, she will say, milk is a poison, you should not drink. So don't listen to all these things, but protein is essential. However, there are young people and some old people who are, let us say, afflicted with a disease and they are recouping in a hospital. To hasten the recovery, you should take protein supplement. There it is good. However, many young people, especially bodybuilders, and nowadays a lot of young people who want to attract girls, they want to build their muscles. And that is possible by taking daily protein supplements. And this protein is usually casein hydrolysate. Sometimes from soya they can take. And therefore, these are essentially milk proteins or soya protein. And they take in large quantities. And that is okay as long as you are young and doing weightlifting and all that, your body looks very nice. But in the long term, these are dangerous. These should not be encouraged. Nobody marries a Mr. Universe. Please remember.
There is a question. Viral enzymes follow the same kinetics?
Yeah. After all, if you're talking viruses, they have only this oncoviruses, they have the reverse transcriptase. Same kinetics, only thing is their proofreading is not very efficient. So the virus makes a lot of mutations it undergoes during replication. That is the problem. Otherwise, whether it is bacterial enzyme or human enzyme or elephant enzyme, they all have the same kind of plant enzyme, microbial enzyme, anything. The only difference is temperature sensitivity. Those who are living in hot sulfur springs, those enzymes are thermostable. Similarly, there are enzymes from Antarctic organisms. They work on only at 4 degrees. If you increase the temperature to 37, they don't work. All these are known, the thermal sensitivity of enzymes. This is a big chapter in biochemistry. But the basic kinetics is all same. There is no difference.
In what way the protein supplements are detrimental in long run?
In the long run, if you supplement with proteins and therefore pump a lot of amino acids after digestion, it will give you muscle protein, but it will affect fat metabolism because there is something called a sparing effect. It will also affect the utilization of carbohydrate, which is the primary source of energy. Therefore, your muscles may become very strong, but suddenly you will see muscle getting arrested. You will die of that shock. It is not good. Most of the weightlifters, they don't live long. They die around 35. That's all. Because they have taxed their muscle, stretched the muscle to such an extent for tone, and that is deleterious. One should be normal. There's no need to become Mr. Universe. Normal health you should have. But protein is essential. Don't avoid protein in the name of religion or in the name of anything. Don't do that. Please take good amounts of protein. And if you are a vegetarian, you can take milk protein. You can drink a lot of good milk, except that in some African populations, after some age, they become milk allergic because they have what is called as lactose intolerance. The enzyme lactase is absent in them after some age. And therefore, when they take milk sugar is called disaccharide lactose. They can't hydrolyze it. And that goes and accumulates in the eye chamber. And you get all sorts of problems with that. Therefore, that allergy, such people which runs in families should not take milk after some age.
Actually, Ms. Jyoti, you can directly speak to sir. I am making you unmute. You directly ask to sir.
Good evening, sir. Very good. Good afternoon. Thank you very much for a very wonderful presentation. Sir, actually, I'm a female, of course, and I am into weight training, and I really Yeah, so I do heavy weights also to keep myself fit. So it's not just the males actually. And although I'm not a pure vegetarian, but I don't eat any protein beyond the protein in eggs. So I do include eggs in my diet. So
There
I
is no
have to
problem.
take,
You
yes,
should
sir.
do isometric exercises. Don't do physical lifting and other kind of gymnasium type. You do isometric exercise is good enough. You become very healthy.
Okay, sir, health is not my only aim. Actually, strength also, I mean, in the sense, I mean, I have no problem in, I mean, I've been doing it for quite a long time, sir. I'm myself a fitness trainer.
Yeah,
I have trained as a fitness instructor,
yeah.
but I just wanted to know if it was okay to take protein powder in moderation. If not, I mean, I don't overdo it. You try to
Absolutely,
see that,
absolutely.
okay,
These are
okay.
available very costly, of course, about 3.5 to 4,000 per
Yes,
bottle
yes, I have
kilogram.
been eating
Yeah,
it. So I have included it in my
then no
diet,
problem.
but
Only thing is
I
do
don't
it in
take much.
limit. Yeah.
Yeah, so I just take one scoop every day in my in milk
No,
or
that is nothing. That is nothing. People
okay.
take one bottle. Don't do
Okay, sir.
that.
No, sir, just in
Couple of
moderation.
spoons, no problem. You will
Okay,
maintain
sir.
your body, and to remain the muscle tone, as we call,
Yes, yes,
you
yes.
can do isometric
Isometric
exercises.
exercise. Sure, sir. I
Don't
will.
do weightlifting and other things. That is not good for the heart. Eh?
Okay, okay.
Okay. And even if you simple do walking, that is good enough for to keep our muscle in tone. But exercise
Yeah,
is essential. If I don't do
I agree,
exercise,
sir.
I become
Yeah.
a tomato, and
Yes.
nobody will marry you.
Yeah, I agree. So I'm not interested in marriage, but
You tell your
yes,
parents I'm not interested.
yeah, they are fine with it,
One
sir.
question, one last question
Thank
from
you.
my side. Sir,
Yeah.
how do we correlate that calculi formation and protein consumption has been there? What about that? Is it a myth or a fact that excessive taking of nitrogenous compounds will lead to the calculi in various parts of the body, like in kidneys or
Yeah, yeah.
can you
See,
highlight?
as I said, our diet, healthy diet, should have enough quantities of nitrogen, carbon, oxygen, hydrogen. Now, in the nitrogenous material like proteins, amino acids, and others, if you take excess, kidney is the one excretory organ. And so you will be taxing because all these proteins are degraded. You get urea, uric acid from nucleotide degradation and all. These are all nitrogenous compounds. And the ability to remove them, excrete them, there is a capacity for the kidney. So if you take too much of protein and too much of other things, you will also get where your joints will have uric acid crystals, and that will cause a excruciating pain. You may have to take tablets for that. And similarly, calculi will be formed in the kidney, Malpighian capsule filters, and therefore it is a very dangerous thing. Of course, this will happen only if you take kilograms of protein every day, which is not, nobody does except those weightlifters who take 100 eggs per day and all that. These are all mad fellows. Don't follow them.
Sir, the after the processing, I mean to say, the digestion will be there and absorption will be there. What could be the rate we will be gaining if we take, suppose, the some quantity suggested, prescribed quantity? What amount of protein will gain? Whether we will gain the, I mean to say, at least 50% of what we are consuming as a supplement.
No, we only know, of course, that's a big area. See, people say that you have to have a diet which has 60% carbohydrate, 20% protein, 10% minerals and other thing, and some fat. If within each category, they will also tell varieties of protein or variety of fat and all. You understand all those essential fatty acids and all those things. Now comes the question what you are asking, what rate of absorption will give me the same proportion in my blood also? That nobody knows, but proteins differ in what they call biological absorption index. There are, for example, plant proteins, you can eat kilograms, it will not contribute to body protein because the biological value is very low. But if you take casein, milk protein, or non-vegetarian food, where the biological index or value is high, almost within that 20% diet, 80% of it can become body protein. So it all depends on what protein you are eating. Same thing for what carbohydrate you are eating. Okay?
Thank you, sir. Thank you so much. There is a question in chat box you might be seeing. Are there any internships regarding protein analysis and enzyme pathology? One participant is asking.
There are laboratories like the Central Food Technology Research Institute in Mysore. There is a Defense Food Institute in Mysore. There are many other labs which will take summer research fellows sponsored by Indian Academy of Sciences. And you can very well go there if you are a student of second year student of BSc or first year student of MSc. You can apply. An advertisement will come in Resonance. I advise all colleges, universities, libraries should subscribe to Resonance. Resonance is a science education journal published by Indian Academy of Sciences, Bangalore. Extremely good articles. Most of your syllabus, you can see articles there written by frontline experts, but in a lucid language. Therefore, please read Resonance as a supplement to textbooks. And in that, you will get an advertisement somewhere in November or October. Apply if you're eligible, because usually we get about 15,000 applications every year, but we offer on about 1,200 fellowships to these students. They are given 6,000 per month. They are placed in a different laboratory in a different part of India. So they understand how